Hello, sir. Am I audible?
Yes. You are very much welcome. If I don't switch it on, on again.
Yes.
If I don't switch it on, it will be okay.
No. I have to switch it on.
Sir, hello. Hello, sir. Am I audible?
You are audible, sir.
Okay.
Wi-Fi.
Wi-Fi right. No. Information. हाँ बोलो. I'm
कॉपी पूरा पास टेस्ट को जो व्हाट्सएप देखिए
संजीव लगे को संजीव सेम DIV है এই তো তোমাকে দিয়েছি WhatsApp
না দিচ্ছি দিয়ে দিলাম দুজনকে
এবার এটাকে দাও সবাই দেখবে এবারে ত্রিবেণীদাকে দেখা যাবে ত্রিবেণীদাকে দেখা যাবে
Welcome back. We now have the third and the last lecture for today by Dr. Subhansu Mondor sir. Let me just introduce him to you. Dr. Subhansu Mondor sir is an assistant professor at the Department of Botany, Bharati University, Shantiniketan. His research interests include microbiome ecology and evolution, molecular biology, microbiome genomics, and proteomics of stress-resistant plant-associated microbiome. From his PhD, he did his PhD in MSc at the Department of Botany, Bharati. He did his PhD at the Department of Microbiology, Bose Institute, and his postdoctorate from the Department of Biomicrobiology, University of Minnesota, USA. He has numerous publications to his credit in journals of national and international reviews and has authored many book chapters. Currently, he is a principal investigator in a program under WDFT entitled Exploration of Cystic Bacterial Community Dynamics of Rice Invertebrate Microbiome from Eastern Himalayan district of West Bengal, a relatively unexplored hotspot for bio-grinding sustainable agriculture. He will be delivering a lecture on cystic microbiome sulfur cycles in anoxic marine cells. So,
Should I start?
hello?
Yes.
Should I start?
Yes, sir. You may.
Okay. So good afternoon, everyone, and I'm very much thankful for giving me the opportunity to represent my work over here. So I am going to present a one of my work like this, which is the. Is it visible? Is it visible?
Yes, it is visible.
Okay. So my today's topic is the microbial. I mean the cryptic microbial sulfur cycle in the marine sediment. So basically, if you consider the sulfur component, this is a very basic diagram of the sulfur cycle and how the sulfur component are recycled through different ecological niches. Like the sulfur dioxide the emission is easily seen from the various type of industrial activity or the volcanic eruptions, and then again it mixed into the atmosphere. It also cause the acid rain, and there are also provision where the different kind of microbes can utilize this sulfur component and it convert into the other form of the sulfur compound like the sulfate that ultimately mixed with the ocean water, and then again a complex cycling, a complex biogeochemical sulfur cycling is actually occurred. So our group is actually working on the sulfur cycle for the last few years. Before going into any details, I would like to share some basic information about the chemolithotroph. So basically, there are organisms that can use the inorganic component as an electron and energy source. Those are known as a chemolithotroph. So there are number of components which can serve as a chemolithotrophic substrate, like the thiosulfate or the tetrathionate, elemental sulfur, sulfide, iron, hydrogen. So there are different group of lithotrophs are there. So what is sulfur oxidizing bacteria? Those bacteria which can utilize the reduced sulfur component as an energy and the electron source. So they are known as a sulfur lithotrophs, and they are taxonomically diverse. They are ecologically diverse. So they have tremendous physiological capacity or tremendous physiological flexibility to utilize the different kind of sulfur component.
Sorry to interrupt you. That your slides are not moving.
Slides will change.
Okay. Can you make it full screen? Yeah.
So is it visible now?
Yes.
So what is the significance for study the sulfur metabolism? So it is the most ancient metabolic process in the earth. Why it is most ancient metabolic process? Because the deepest phylogenetic root or branches can occupy by hyperthermophilic sulfur lithotroph. And if we improve our understanding regarding the sulfur lithotrophy, it will ultimately unreveal the primitive metabolic signature or primitive metabolic strategy of early life. So these sulfur oxidizing bacteria can occupy a diverse range of habitat. They can present in different kind of extreme habitat as well as in some normal habitat also. Like if you consider the acid mining lineage, there is a very important signature bacteria is present like the Acinetobacillus bacillus. If you consider the geothermal hot spring, then sulfate hydrogenabium is a signature bacteria. The sulfolobus present in the hydrothermal vein, or the thiobacillus, they can present in the perennially ice-covered Antarctic lake. So they can occupy a extreme habitat. They also capable to reside in some other habitat also. So there are there are three known mechanisms of sulfur chemolithotrophy. One is the alpha photobacterial SOX pathway, as I already told that there are different reduced sulfur component like the thiosulfate, pentathionate, elemental sulfur, sulfide. So all these components can act as an electron and an energy source for the bacteria. So these sulfur lithotrophic bacteria are phylogenetically diverse. and they have diversified metabolic pathway to utilize the different kind of sulfur component. Like in case of alpha bacterial SOX pathway, where the high sulfate is directly oxidized into the sulfate without formation of any detectable intermediate. Like most of the members of alpha bacteria are follow this kind of pathway. Whereas the Brown's thiosulfate oxidation pathway, where the thiosulfate converted into the sulfate by the formation of elemental sulfur as a potential intermediate. So this kind of pathway can be seen in the members of chromaceaceae or the ectothyroidospiralaceae. There is, as an example, we can see that is the allochromatium vinosum is a very common example who can follow this kind of Brown's thiosulfate oxidation pathway. However, the last one is the S4I pathway, that is the tetrahydrocannate intermediate pathway. This tetrahydrocannate intermediate pathway, in this case, the thiosulfate oxidizes into the sulfate via the formation of hello. Slide change করছি এখন তো একটা স্লাইড উপরই বলছি শোনা যাচ্ছে Okay. So these tetrahydrocannate form as an intermediate during the oxidation of thiosulfate. So there are a number of bacteria like the Tetrahydrocannate cascinaceae that actually isolated by my PhD supervisor during his PhD tenure. And I also worked on some other sulfur bacteria which we have isolated from some extreme environment like the geothermal hot spring as well as the Arabian Sea OMZ. However, in doing so, like before going into any details, I would like to share some basic information about the metagenome. Because we all are working with the microbial ecology of the extreme habitat, and there are number of organisms. There is a huge population of bacteria present in the extreme habitat that we cannot isolate using our conventional pure culture based approach. So basically, here is the utilization of the metagenome. So what do you mean by metagenome? Metagenome is nothing but a total environmental pool where the pure culture dependent isolation approach has been proven to fail. And that case, for the devolution of bacterial community architecture, or if we want to study the structure function relationship of any particular environment, then we can take the help of the metagenome. Simply, we can isolate the entire DNA that include the bacteria, that include the virus, that can include the other organism, but we only work with the bacterial DNA. So we isolate the metagenomic DNA, just like the genomic DNA isolation. Then we send for the sequencing. So there are number of sequencing platforms are available on which we can do these things, like the Illumina or the Iron Torrent or the PacBio. So these are the different sequencing platform on which we can sequence our metagenomic DNA. So after sequencing, we get the rich RODIT and we have to assign. I mean, this is the taxonomic assignment. Then we have to classify their metabolic type. We can also predict their metabolic function. So in this way, we can analyze the entire bacterial community architecture, their structure function relationship, and some other type of function, and their probable role on which they reside. And there are some databases are also available, like the RNA, there are NCBI databases available, Silva databases are available. There are a number of genome databases are also available, against which we can do these all kind of bioinformatics. And this is a very important tool nowadays, because without the help of the metagenomics or without the help of genomics, we cannot explore the cryptic structural, I mean the bacterial community structure in the in a particular environment. So in this case, I mean in this particular study, our main objective to find out the potential involvement of tetrahydrocannate in the sulfur cycle of anoxic marine sediment. So why tetrahydrocannate? Because among the reduced sulfur substrate, tetrahydrocannate, the concentration of the tetrahydrocannate is very low and it is very unstable in the nature. So it is very difficult to detect the tetrahydrocannate in the natural environment. So there is a very less amount of study available on where they can explore the exact role of the tetrahydrocannate in the marine sulfur cycle. So our main motto that what is the exact tetrahydrocannate metabolizing bacteria are present in the sedimentary microbiome, what is their role in the global sulfur cycle. So that is the major objective of this particular work. However, this is some basic information that how we did all these things. Like our group also have a collaboration with the National Institute of Oceanography, Goa, from where we can collect the samples. So I will show you how we collected the samples. So initially we take the two different approach, like the culture independent approach, and second one is the culture dependent approach. So in case of a culture independent approach, the community DNA and the RNA is actually extracted. So that is nothing but the isolation of the metagenomic DNA. Then we run our DNA in a specific platform. We get the read and we assemble it, we annotated it, and we and classify up to their lowest taxonomic level, and on which the entire population genomic study is actually based. And the second one is the culture dependent approach. At the same time, we also attempted to isolate a number of tetrahydride metabolizing bacteria from these particular environment. We isolate them, we identified them using the 16S gene sequencing approach, and then we characterize at different physiological level that what are the different sulfur substrate they can form, what extent they can form, what is the different kind of intermediate they can form during the oxidation of the tetrahydride or during the oxidation of the thiosulfate. So now I welcome all in the world of microbes of ocean. Just before going into the sample details, I would like to share some basic information about the OMZ. So what are the OMZ? So OMZ is nothing but a oxygen minimum zone of any marine environment. It is not only restricted within the Arabian Sea, but also there are a specific OMZ zone are present in the Namibian Ocean or the Eastern Tropic South Pacific Ocean. So usually they form in the very poor ventilated ocean water, where the respiratory oxygen demand is high for the organic matter degradation. And the main reason for the formation of the OMZ is a global warming. So global warming is a very alarming thing, which ultimately generate a oxygen-less environment in the ocean system. It will impact the entire biodiversity in the ocean system. So our major objective of this particular work is to find out the exact role of the tetrahydride in these anoxic.
Your slides are not moving.
I didn't move the slide till now. Is it moving?
It's not moving now.
Is it moving now?
Slide. You could try it full screen. It's moving now. It's moving.
Moving.
It's moving.
So you can see the picture of the OMZ globally.
Yes.
Okay. So that is the geographic location of the ASOMZ sampling point from where we have collected the sample. So as I already told you, this is the sampling strategy, advanced sampling point. We have a collaboration with the National Institute of Oceanography, from where we conduct a cruise that is known as the RV Sindhu Sankal. And how we collected the sample, that is suppose this is the RV Sindhu Sankal, that is a ship, and the entire cruise is conducted by the NIO Goa, and we collected the sample from these particular 2 points, which lies at the center of the OMZ. That is the SSK 40, named as SSK 42.5 and SSK 42/6. So one is the 530 meter below the sea floor, another one is the 580 meter below the sea floor. So in this way, we collected a 3 meter long core, and during the collection of the core, after the collection of the 3 meter long gravity core, we cut the sample at 15 centimeter resolution to collect the sedimentary microbiome, or to perform the sedimentary microbiome analysis. So under a continuous nitrogen shower to maintain the anoxic environment. So this is the schematic diagram which shows how we maintain the anoxic environment and during the collection of the particular core. So there are 3 meter long core, and at every 15 centimeter resolution we did the metagenome. So in this way, these are the some sample details. I am not going into the statistics or some other details. However, there are points like the first point SSK 42/5 range from the 0 to 295 cm basic centimeter below the sea floor, and in every cases we perform the metagenome twice, uh, and then we analyze the structure of the community DNA. So this is the basic statistics, and in every case we perform the duplicate metagenomic sequencing for the analysis of in situ microbial community. And this is for the SSK 42/6, where the sediment depth range from the 22 to 275 cm base. So this is the basic data for our metagenomic analysis. So what is our main focus is that we categorize the entire bacterial community into three particular types. One is the tetrathanate forming bacteria, second one is the tetrathanate oxidizing bacteria, and third one is the tetrathanate respiring bacteria. So tetrathanate forming bacteria means which can form the tetrathanate during the oxidation of the thiosulfate. Just like the marine strain of all the marine strain of Pseudomonas and Halomonas, they all are capable to form the tetrathanate during the oxidation of the thiosulfate. So they are considered as the tetrathanate forming bacteria. And we specifically categorize or specifically find out what is the relative abundance of the Pseudomonas, what is the relative abundance of the Halomonas, how they are distributed down the sediment core. And second category is the tetrathanate oxidizing bacteria, because our main focus or the major objective of this particular work is to find out what is the exact role of the tetrathanate in the sedimentary microbiome. So as a tetrathanate oxidizing bacteria, there are three bacteria which are obligately tetrathanate oxidizer. They are the Acidithiobacillus, second one is the Halothiobacillus, and third one is the Thiomicrospira. So these three bacteria are capable to oxidize the tetrathanate into the sulfate, and they also can form the tetrathanate as an intermediate product during the oxidation of the thiosulfate. And this particular figure A and B shows the correlation analysis among these tetrathanate metabolic type. And the third one is the tetrathanate respiring bacteria. That's like the Salmonella, Cyclobacter, Proteus, etc. So in doing so, we also perform the pairwise Pearson correlation coefficient analysis, or we can say it is a simple Spearman rank correlation coefficient analysis, and that is calculated to estimate the interdependence between the environmental parameter or the diversity or prevalence of bacterial group in individual sediment core. So in this way, we have found that these blue circle is actually indicate a strong positive correlation, whereas the black circle shows the no correlation or the mutual or I mean just simply a neutral value denoted by the black circle. So what we have found is that there is a strong interdependency between these three metabolic types. So these analyses directly indicate their strong syntropic relationship. I mean they are interdependent in terms of their metabolic substrate, in terms of their ecological lifestyle. So these tetrathanate forming bacteria, tetrathanate oxidized, we categorize these all entire bacterial community into three particular metabolic types. So these Salmonella, Cytobacter, and the Proteus, they can be considered as the tetrathanate respiring bacteria. So what is the product of the tetrathanate respiration? That is the sulfide. So during the oxidation of the tetrathanate, during the respiration of the tetrathanate, they reduce tetrathanate into the sulfide. So in this way, we find out that what are the major groups that are present in the sedimentary microbiome and how we can denote them, and we can classify them based on their metabolic substrate preferences. So next, we have performed the slurry culture analysis. Just like the first figure, this first figure is actually shows the tetrathanate formation rate in a slurry incubated sample. That means we also find out that whether all these bacteria which we get from the metagenome is really alive and active in situ condition. So we have found that when we perform our slurry culture experiment, we have found that there is a specific tetrathanate formation rate that is and in both the studied code, SSK 42/5 and SSK 42/6. This SSK 42/5 is nothing but a name of a 3-meter long pore which is present at a 530 meter below the sea level, whereas the SSK 42/6 that is present at the 580 meter below the sea level. So in this way, we check their metabolizing capacity and whether they are prevalent and active in situ condition. Because the metagenomic data only give us the idea about the entire bacterial community architecture, not about that exact functionality until we perform the transcriptome or the proteome. So at this level, we have checked that tetrathanate formation rate is there, and figure C and D where the violet line indicate the tetrathanate oxidation rate, and the third one that is tetrathanate reduction rate. And we have found all the slurry incubated samples, the tetrathanate formation, tetrathanate oxidation, as well as tetrathanate reductions are present. So in this way, we get to confirm, okay, that all these bacteria, I mean, what we are thinking about about the tetrathanate forming bacteria, whether they are oxidizing, whether they are respiring, all they are metabolically active. Not only present in the metagenome, but also they are present in the sedimentary microbiome or the anoxic marine sediment. Now, our major role, what are the potential candidates that we can isolate from these sediment? We have isolated a number of bacteria. We have identified them in a different media. Like the first one is the halothiobacillus. It is a very common sulfur oxidizer, and we have isolated from these SSK 42/6 in the ASWD media, or we can say it is the artificial seawater media. So there are certain media that are specifically designed for the isolation of marine bacteria, particularly marine sulfur bacteria. So ASWD or artificial seawater medium is a very common media, and it is routinely used for the isolation of marine sulfur oxidizer. So we have isolated a number of bacteria: Halothiobacillus, Methylophaga, Halomonas, Pseudomonas, Strenotophomonas, Pusillimonas, Enterobacter, and most interesting part is that every bacteria have the physiological flexibility to utilize the tetrathanate in a different way. I mean, some are utilized as an electron and energy source. Some are utilized as a respiratory substrate. Some can form the tetrathanate as an intermediate during the oxidation of the thiosulfate. So these are the some details. I mean, these are the some strain name, and these are the some phenotypic details that what they actually perform during the physiological study. And this is the NCBI accession number, because we sequence all the genome of these isolates. So there are also provisions that to find out that what are the actual relative abundance of these particular bacteria in the sedimentary microbiome, because we have the metagenome in our hand, we have the whole genome sequence in our hand. So now we can perform that what is the exact relative abundance of these particular candidates in their native environment. So next we perform the physiological study, like these halomonas these halomonas, these Methylophaga, Pseudomonas, Strenotophomonas, all these 4 bacteria can oxidize the tetrathanate, can form the tetrathanate as an intermediate. So they are designated as a tetrathanate metabolizing bacteria, and they can participate in the thiosulfate to tetrathanate conversion. All these four bacteria, whereas the E and the F can be designated as a tetrathanate oxidizing bacteria, because they can convert the tetrathanate into the sulfate. And the last one, the Enterobacter, is a tetrathanate respiring bacteria, because they can reduce the tetrathanate, and as an end product they can form the sulfide. Okay, so in this way we have checked their metabolic potential. I mean, what is the exact sulfur oxidation pathway that they follow during the oxidation of reduced sulfur component. Here our major focus is the tetrathanate, so we only restricted our study on the tetrathanate metabolism. Next, we sequence as I told you that we have sequenced the whole genome of all these isolates to know about their more genetic machinery, that what is the exact genetic determinant, what are the genes that are responsible for the tetrathanate metabolism, how they oxidize the tetrathanate, how they reduce the tetrathanate, what is the end product, and what are the other features that present. And this table is actually is nothing but the demonstration of the basic genomic whole genome statistics that we have mentioned here. So I'm not going to read out all these things. So the next point is to find out that what are the percentage of metagenomic reads from the individual sediment samples. Like what we did in we actually mapped the whole genome onto the metagenome using the Botany. So in this way, we successfully find out that what is the exact percentage of these bacteria that are present in that particular sediment. I mean, we have at least 12 to 13 points. You can see that range from the 0 to 295 centimeter, and at every point we have the metagenome. And what we have did, at every point we map our whole genome sequence onto the metagenome and find out what is the matching percentage. So in this way, we successfully find out that all these bacteria are actually present in the sedimentary microbiome on which the present study is based. So this is the pictorial demonstration of our mapping experiment of where we actually this black circle is actually denoted the percentage of metagenomic reads that actually match with the genome sequence of an isolate. You can find these are the two important code on which the present study is based: SSK 42 by 5 and SSK 42 by 6. And these are the abbreviation of the bacterial isolate: the Enterobacter, Halomonas, Halobacillus, Methylophaga, Fusilimonas, Pseudomonas. And you can see at every depth, at every sedimentary point, these bacteria are present in a very significant amount, whether it is SSK 42 by 5 or whether it is SSK 42 by 6. So now we can assume that they might play the role. They might play the role in the sulfur cycle that entirely depend or entirely focused on the tetrathanate. But as I already told you, that the metagenome can only give us the idea about the community architecture, not about their functionality. We can predict the functionality. We certainly predict the functionality of any metagenome, because there are provision to find out the presence of the genes or the enzyme, or there are also provision to assemble the genome from the metagenome. That is known as a metagenome assemble genome (MAG). So we can do it, but to find out the exact function, we need to perform the transcriptome. So this is that's why we performed our metatranscriptome analysis from a particular point, that is the 275 centimeter below the CMBSF or centimeter below the CPRO of the SSK 42 by 6. Then we sequence the transcriptome using the high-sec Illumina sequencing. Then we use our all these raw data against the Silva database. That is a very common database for our metatranscriptome, or it is a very common RNA database. So what we found from the metatranscriptome. The bottom line is that we have found, or we have detected, a number of genes that are responsible for the oxidation of the tetrathanate, like the SOXY. It is a very well-known gene in the domain of sulfur bacteria. Like it is a very important gene which directly participates in the conversion of the tetrathanate into the sulfate. And we also able to detect the organism source from which the gene is actually detected. So in this case, we have detected the SOXY is derived from the acidithiobacillus. So not only the SOXY, the mRNA study, that metatranscriptome study also shows the presence of the high sulfur reductase gene that usually came from the Salmonella. So that is a potential tetrathanate respiring bacteria. So in this way, we find out the presence of potential gene. So this particular table is actually showing the name of the genes. Like this is the TSDA. The full form of the TSDA is the thisulfate dehydrogenase, and the major function of this particular gene is to convert the thisulfate into the tetrathanate. And these are some organism source of the closest state. So not only the TSDA, we have also detected the SOXB, that is the thioesterase, that also directly involved in the conversion of the tetrathanate into the sulfate. We also detected the SOXY, as I already told you. So in this way, we completely analyze the metatranscriptome of the 275 cm base pair, and we find out that the potential genes that are thought to be involved in the tetrathanate metabolism or tetrathanate conversion that are present in the sedimentary microbiome. So now we can confirm that these bacteria are alive, they are active, and they can participate in the tetrathanate metabolism in a different way. So next, our major target is to find out the exact role, exact concentration of the thiosulfate, because thiosulfate is a very important intermediate product of the any kind of sulfur cycle, whether it is a marine sulfur cycle, it doesn't matter whether it is a terrestrial sulfur cycle. So it is a very important product because most of the sulfur lithotroph can utilize the thiosulfate as a electron at the energy source. So it is a it is a very common substrate that can be utilized by almost all type of sulfur lithotrophs. So these are the some data where we got from our collaborator NIO, and it shows the these violet circle is actually shows the presence of the sulfate down the sediment depth, and these brown circle is actually showing the presence of the sulfide, and the blue one shows the presence of the thiosulfate. So now we geochemically detected the presence of the thiosulfate, tetrahydrosulfate, sulfate. Biologically, we have detected the presence of a number of bacteria that are capable to metabolize the tetrahydrosulfate in a different way. So now our last target is to construct a complete sulfur cycle that thought to be operational in this particular anoxic marine sediment. That's why we constructed this particular diagram, that these are the potential biotic or the abiotic process that are involved in the formation or transformation of the tetrahydrosulfate. So let me explain this sulfur cycle model. Here, in this case, the tetrahydrosulfate, consider the tetrahydrosulfate as a center product. This tetrahydrosulfate, what is the next? The first question is that what is the source of the tetrahydrosulfate in the anoxic marine sediment? So there are lots of geochemical provision by which abiotically tetrahydrosulfate can form. Like if you consider this particular thing, this particular scheme, you can see pyrite. It is a very common component in the marine sediment. Pyrite, in the presence of the manganese dioxide, can abiotically convert into the tetrahydrosulfate, and it can also form the thiosulfate. So these are the some probable source of thiosulfate and the tetrahydrosulfate in the marine water as well as in the marine sediment. Now this tetrahydrosulfate can be converted into the thiosulfate, or we can say can be reduced into the thiosulfate by the activity of bacterial group C, which are considered as the tetrahydrosulfate respiring bacteria. So during the reaction, they can form the sulfide as the end product. And sulfide, again in the presence of the iron oxide, it can combine with the iron oxide and it can again regenerate the pyrite. So pyrite ultimately again generate the tetrahydrosulfate and the thiosulfate. So the thiosulfate again can convert it into the sulfate by the formation of tetrahydrosulfate by the bacterial group A. What is the bacterial group A? The Pseudomonas and the Halomonas. Both these bacteria are very much efficient for the formation of the tetrahydrosulfate from the thiosulfate, because we have isolated lots of strain of Pseudomonas, lots of strain of Halomonas. All isolated bacteria are capable to oxidize the tetrahydrosulfate into the oxidize the thiosulfate into the tetrahydrosulfate. Now this tetrahydrosulfate can directly oxidize into the sulfate, and it can be utilized by a specific group of bacteria that is designated as a bacterial group B, that is the Acidithiobacillus, Thiomycosphaera, and the Halothiobacillus. So they are considered as a obligate tetrahydrosulfate oxidizer on which they can form the tetrahydrosulfate into the sulfate, and this sulfate again form the tetrahydrosulfate, and this particular function is actually carried out by the other sulfur reducer, like the disulfovibrio or the disulfobulbus. So the significant portions of the sulfur reducer are also present. Present. So in this way, these sulfur component, these thiosulfate or the tetrahydrosulfate can interconverted biologically by the activity of diverse group of microbes, as well as abiotically by different geochemical pathway. So this kind of tetrahydrosulfate based pathway, the information regarding the tetrahydrosulfate based sulfur cycle in anoxic marine sediment is relatively little, or I can say it is a very scarce information, because tetrahydrosulfate is a very difficult thing to detect in the natural environment. So all these data is actually published in our European Geoscience journal, Geoscience. the role of tetrahydride in the sulfur cycle. So thank you, and again thank you for the organizing committee for giving me the opportunity to represent my work here, particularly Dr. Bosu Sarvadhikari. And if you have any question from these, I'm glad to answer you all.
Thank you very much, Subhranshu sir, for your beautiful lecture. We came to know about the sulfur cycles of oxidizing, reducing bacteria in marine sediments. Your lecture, as far as I could understand, was about the role of tetra. Can you hear me?
Yes, sir.
Okay. Was about the tetrahydrogenate, its role in the sulfur cycle, and how bacteria are involved in that conversion of that into different sulfur compounds.
Yes, sir.
So I have opened the session for question. If any question anybody can ask. Do you have any question? Very nice question. One question has been asked by Shruti Nayak, our botany student. She is asking, what are the implications of this cycle or this research in the current environmental situation?
Okay.
I mean,
very nice question. So let me tell you one thing, that as I already told you, that this sulfur metabolism is a ancient and a very primitive metabolism. So because the deepest rooted phylogenetic tree are occupied by hyperthermophilic sulfur lithotroph. So it is one of the approach to study the early metabolic strategy of early life. If we can able to study this kind of metabolism, we can stress out, we can understand, we can improve our understanding regarding the exact type of primitive mechanism that thought to be operated in the early life or in the primitive earth. So that is the thing for which we particularly interested to study the sulfur cycle, mostly in the anaphytic sediment, because at the starting of the earth, at the primitive cycles, there is no oxygen. So mostly those organisms which are present, they use nitrate or the reduced sulfur, the elemental sulfur or the sulfide as a terminal electron acceptor, not the oxygen. So that is the one of the way by which we can study the primitive metabolic strategy of early life.
Okay, sir. And one of my question is, during your did you find any acid mine drainage kind of H2SO4 synthesis was there anywhere or in this
Yes,
cycle?
sir. Yes, sir. There are a lot of bacteria where the end product is the sulfate, and we have detected a significant portion of significant presence of acidithiobacillus. So usually this acidithiobacillus is a very signature bacteria that usually present in the acid mining drainage. But surprisingly, we also detected this particular bacteria in a high percentage in the sedimentary microbiome. So maybe there is nothing but a mixing of the industrial water in the ocean sediment for the long time, and that's why this kind of bacteria also can be find out in this type of anaphytic marine sediment.
Another question is, do you have any question, student? Anyone? So I can ask one question. Do you have question, sir?
Sir, could you please repeat?
I'm asking one more question.
Yes.
As far as the great oxidation event. Hello.
Sir, we are actually the detection of the oxygen is also tried out, because this particular portion, the OMZ, oxygen minimum zone, is actually formed for the long time due to the global warming. So our collaborator, Dr. Anindo Mujumdar, his team is also trying to find out the stresses of oxygen, and they're not able to detect a detectable amount of oxygen despite having a lot of effort there. So there is a sequential work that is actually published in the famous microbiology letter from our group, that how the life is actually possible in the anoxic marine sediment. Because if you see meticulously, sir, you will get to see there are bacteria in the metagenome that are known as aerobic bacteria. But what is the source of this cryptic oxygen? So we postulated a hypothesis that there are perchlorate. It is a common component in the marine sediment. Perchlorate is actually responsible for such for production of a very minute amount of oxygen, and that can be efficiently utilized by the native microbiota. And that is the separate work, sir, and that are already published in another journal.
Okay, so thank you, and hope this lecture will help our students to. Asha sir, did you able to culture any organism from this?
Yes, sir.
Microbiota.
Yes, sir. We have, as I already show you, that we almost isolated 10 to 15 bacteria from different metabolic types. I mean, we have successfully isolated the BHALOTIA bacillus, Pusillimonas, Methylophaga, Pseudomonas. So all these bacteria isolated from this sediment, from this anoxic sediment, using the culture-dependent approach. We have identified, we have sequenced the genome of this isolate also.
Thank you so much. Thank you so much. So now we will go for our valedictory session is there, but before that there is a poster session. I would request you. How do I don't know. How do you be there? Because 10 minutes will be required for. Hello.
हाँ sir.
Are you hearing me?
Okay.
What I'm saying that our lecture sessions have finished. Okay, and now we will go for a valedictory session, but there is a 10-minute break for poster presentation, judgment or evaluation. So
Okay.
I would like you to stay during the valedictory session, if it is
Okay.
possible. You may take a 10 minutes, leaving the net on, and then you come back after 10 minutes here, and we will focus the webcam towards the valedictory person who will be giving the vote of thanks towards that. So you can enjoy the vividness of this lecture hall too.
Okay.
Am I right, sir?
Okay. It's not a problem.
Okay. So 10 minutes break for you, and then you come back again, please. Thank you so much. Okay, just wait for 5 minutes.
So should I leave now? I will join after 10 minutes. হ্যাঁ ঠিক আছে, তাহলে আমি এখন অফলাইন হয়ে যাচ্ছি।
ভিডিওটা অফ করে আর মিউট করে কি অফলাইন।
ঠিক আছে, ওকে।